At the request of several fanciers in matters of inbreeding, it appears that most fanciers do not know the subject and often sow confusion. In this video, we will try to clarify in detail inbreeding in pigeons. Unfortunately, it is not possible to start the debate directly without first having introduced some notions of biology for the good comprehension. We will cite as an example plants and animals. They are made up of cells. Each cell contains a nucleus which is immersed in the cellular environment. In the nucleus, there are very special filaments which are called chromosomes and which carry in a determined order the genes which contain the chemical material involved in the transmission of hereditary characteristics. Genes are the units of heredity. Each animal species has a determined number of chromosomes. The pigeon has 80. The chromosomes are 2 by 2, so the pigeon has 40 pairs of chromosomes. Each pair of chromosomes has a characteristic appearance and shape: elongated, short, thick, thin, stick-shaped, or curved, so that a specialist can recognize them. The two chromosomes in a pair are called homologous chromosomes. If they have the same external appearance, they are not necessarily identical. Indeed, the genes that a chromosome supports can be different from those of its counterpart from a purely genetic point of view. The alternative forms in which a gene can appear are called alleles. The starting point of an organism is made up of a female cell, the ovum fertilized by a male cell, the sperm. This fertilized egg will split into 2 daughter cells, then later into 4, and so on until another creature is created. Before a cell gives rise to 2 daughter cells, each chromosome will divide lengthwise into 2 equal parts. This is accompanied by a split of each gene. This basic process occurs during each cell division, so that each body cell of the pigeon will have the typical 80 chromosomes. Inbreeding is considered by many fanciers to be a shameful disease, indeed, very poorly defined, but which often serves to explain or justify the many problems and failures encountered in pigeon breeding. It is therefore useful to demystify this notion, very often misunderstood and confused with something else. In the genetic sense of the word, consanguinity is the culture between members of the same family. If we cultivate between members of the same family, it is because they all have qualities that we seek to reproduce. We will therefore cultivate an inbreeding of family where there are many good pigeons, so as to try to increase the percentage of good pigeons revealed each year. But the characters of each pigeon are multiple. There are those that we see, feel. It is the phenotype, such character of the skeleton, long, short, deep, flat, etc., of the plumage, color, shape of the remiges, thickness of the cover plumage, of the eye, etc. There are hidden characters, recessive, which can reappear when they also exist in the spouse, hidden, that is to say recessive. This is the genotype. And then there's what we're all looking for, sporty quality. We see that, we try to explain it, but it's not obvious. All the theories that have emerged throughout the history of pigeon racing all aim to allow the detection, a priori, of the right pigeon. All the art of inbreeding will therefore be to mate together pigeons with similar physical characteristics, equal sporting value, and having a common origin. Indeed, many hereditary traits are multiple genes. For example, the color of the iris. One might think that the character red eye is a simple character and that, when we mate a red eye with a white eye, we draw young with red eyes and young with white eyes. It's much more complicated than that. In each basic color, either brown, yellow, red, white, there are several genes, hereditary character, as the brown dominates the yellow-red, which itself dominates the white. No pigeon has a pure eye. For example, a red eye contains not only at least one yellow character, but also perhaps a white character. Hence the extreme variability in the color of the eyes of the descendants, depending on whether there are more or less yellow characters compared to red, and white characters, recessive. One can only say that there is pure color when the eye is white, even though this eye is more or less sandblasted with pink. We see the extreme complexity of the case. Let us add that in macaws the iris is discolored in proportion to the white feathers, and then appears partially black. In fact, it is discolored, and it is the retina black that we see in the back of the eye. Mating brother and sister, on the absolute condition that both are excellent racing pigeons, gives a very good proportion of good pigeons, perfectly normal. These pigeons from brother and sister are also good breeders, especially coupled with a spouse too, descended from brother and sister of another origin. Each pigeon fancier wants to improve the quality of his colony by discarding the unfit birds, sort, and by choosing his future breeders as well as possible, select. By this method, he tries to fix the favorable characteristics of his favorites. First of all, it should be noted that the good characteristics are not the same for everyone. Thus, there are many pigeon fanciers who like above all to have a colon, i.e. uniform in shape and color. The latter are inspired by what happens for fancy pigeons. It is indeed a relatively easy task to fix. Determine characters such as the color of the plumage, see above the example of blue and black colors, and to quickly arrive at a certain uniformity. They forget that we are dealing with carrier pigeons. When it comes to quantitative characters, this is the case with the carrier pigeon. We come up against the difficulty of recessive genes. The latter only appear if the genes are present in the double state, homozygous, one from the father and one from the mother. This is easier to achieve by mating related subjects, since these are more likely to be endowed with the same factors. Now, if we take into consideration that the recessive factors are mostly unfavorable, we better understand the dangers linked to the practice of the length of the wing, etc. The improvement of the results in the competitions which interests us the most. Is it possible to obtain added value from the carrier pigeon? In principle, yes, but it is enough to be convinced of it to think of the financial means, which it is necessary to have, as well as the space necessary for the implementation of such a project. I believe that even the huge facilities currently available to some pigeon fanciers, with several thousand pigeons, could not be enough to hold several pure lines crossed. Then between them, add to this the difficulties of limited multiplication. It is not possible to breed pigeons in chains. There are, however, unmistakable indications. These are the pedigrees of national and international winners or other long-distance champions from crosses. I am convinced that it is not possible to give recipes on how to proceed to achieve the goal. What equipment to take at the start? What are the limits not to cross in the practice of consanguinity? This is always the case when everyone's situation is different. This is how everyone, for example, has a predilection for a game system or for a certain distance. Confidence in the pedigrees is indeed very important. It allows to check if the pigeons come from multiple crosses, or if they belong to a noble family where the chances of bringing out favorable combinations are higher. In addition, I would like to insist on some important points, especially for those who have to work with limited means. 1. To form a colony or during a new introduction, the basis should preferably be acquired from serious, trustworthy fanciers who are specialized in the same type of game that is envisaged. Better pigeons from 2 lofts than birds from many fanciers. 2. Don't be seduced by the past glory of the big names. 3. Anyone who can afford to buy a winning pigeon with a view to putting it into production should also know that his pedigree is as important as his price list. The family tree must mention more than the sire and the mother. It must cover several generations. 4. It is important not to play purchased animals which must serve as a basis and wait to have a certain number of descendants before carrying out tests. 5. At the beginning of the formation of the colony, it is necessary to basket the bred youngsters, especially the females, often at the races and not be satisfied with racing a few well-prepared ones on 1 or 2 stages. They are therefore all placed in the same circumstances, which makes it possible to make a valid judgment in order to select them. When making this selection, one must always keep the results in mind and not allow oneself to be influenced by details. 6. You have to be very careful with close inbreeding and prefer a family breeding in a closed system, allowing, however, the limited introduction of new subjects over the years. If possible, always try to introduce pigeons that have some common blood with the good sailboats you already have. When a crossover combination works this well, there's no doubt something special happened. Specific gene pairs are of such high quality relative to others that they tend to dominate. This is called super dominance or epistasis. This exceptional effect only occurs in very rare circumstances. There are very few known examples where these repeated combinations have worked so well and produced many champions. This needs to be cultivated very carefully because success at this frequency and at this level requires a little more than just luck. To finish, don't be afraid of inbreeding anymore, but learn to use it.